and welcome to Sam Biology class. Friends, today we shall discuss about Griffith's experiment. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Griffith's experiment was the first experiment to demonstrate that bacteria are capable of transferring genetic information through a process called transformation. This experiment was conducted by British bacteriologist Frederick Griffith. Pneumonia was a serious cause of death in the wake of post-World War I Spanish influenza pandemic. Griffith was studying the possibility of creating a vaccine. In his experiment, he used two strains of Streptococcus pneumoniae. Streptococcus pneumoniae is a lancet-shaped gram-positive bacteria. It is the main cause of community-acquired pneumonia and meningitis in children and elderly, and that of sepsis in individuals infected with HIV. Out of two different strains of Streptococcus pneumoniae used by Griffith, one strain was a type 2R bacteria and the other strain was a type 3S. Type 2R bacteria were devoid of the protective polysaccharide capsule. Absence of capsule gave type 2R bacterial colonies a rough appearance. Hence, type 2R strain is also called rough strain. And here R means rough. It is because of the absence of capsule. Type 2R bacteria were destroyed by host immune system through phagocytosis. Because of this, type 2R strain is non-virulent. So when type 2R bacteria were injected into mouse survived. By contrast, type 3S bacteria had the protective capsule. Presence of capsule gave type 3S bacterial colonies a smooth appearance. Hence, this strain is also called smooth strain, and here S means smooth. Because of the presence of polysaccharide capsule, type 3S bacteria were protected from phagocytosis, as a result of which they escaped host immune system and caused pneumonia. So type 3S strain is virulent. Because of this, when type 3S bacteria were injected into mouse died of pneumonia. In another experiment, Griffith injected heat-killed type 3S bacteria into the mouse, and he found that mouse survived. It is quite obvious because the bacteria has been killed by heat, and heating has destroyed its virulence, because of which the mouse survived. But surprisingly, when Griffith injected a mixture of live type 2R bacteria and heat-killed type 3S bacteria into the mouse died. And Griffith was also able to isolate both live type 2R and type 3S bacteria from the blood of those dead mouse. So the finding is interesting because when we inject type 2R bacteria alone into the mouse survive. This is because the strain is non-virulent. And when we inject heat-killed type 3S strain into mouse survives. This is because the bacteria has been killed by heat. But when the mixture containing live type 2R bacteria and heat-killed type 3S bacteria, mouse died of pneumonia. Based on this finding, Griffith concluded that type 2R bacteria was transformed into lethal type 3S bacteria by a transforming principle which was somehow a part of heat-killed type 3S bacteria. So this is the process of transformation by which a specific component has moved from heat-killed type 3S bacteria to type 2R strain and converting it into lethal type 3S bacteria. But Griffith was not able to identify the exact nature of the transforming principle. Now we know that it is DNA. By the process of heating, type 3S bacteria were killed, but their DNA survived the heating process. So when this mixture was injected, DNA from heat-killed type 3S bacteria was taken up by type 2R bacteria, as a result of which type 2R strain was transformed into lethal type 3S strain, which caused pneumonia, as a result of which the mouse died. And after that, the exact nature of the transforming principle was verified experimentally by Avery, M.C. Lloyd and M.C. Carty, and by Hertz and Chase. To know more about transformation, please watch my other videos. So friends, this is all about Griffith's experiment. For more updates, please subscribe my YouTube channel, Sam Biology Class.